Hi, in this video, we'll talk about enzyme kinetics and regulation. So the objectives are for you to be able to interpret basic rate versus substrate concentration curves for enzyme reactions, to list common ways that enzymes are regulated, and to recognize the effect of a competitive inhibitor on rate versus substrate concentration curves. Okay, so this all starts with the fact that the enzyme catalyzed reactions occur at enzyme active sites. And the very first step is the enzyme binds the substrate or substrates in the active site. Okay, so reviewing from the previous video about enzyme catalysis, we're looking at delta ALA synthase active site pocket here, right, with the substrates all bound. So that's the very first step of the reaction. And only after the substrates are bound on the enzyme active site can products be formed, right? And the reason this is important, the fact that the enzyme first must bind a substrate at its active site before you see any product, that gives us what's called saturation kinetics. So let me show you with this curve down here. The types of curves we'll be looking at in this video all measure the rate of reaction or reaction velocity on the y-axis versus the substrate concentration on the x-axis. And what we see here is when the substrate concentration is relatively low, binding of the substrate is rate limiting for the reaction. So that means that at low substrate concentrations, when we increase the substrate concentration, we see a big increase in reaction rate, right? But eventually, when you add more substrate, you no longer see that big increase. It saturates because all the available enzyme active sites are bound with substrate. So providing more substrates not going to make the reaction go any faster. In this range, the rate of the reaction is limited by the rate of chemistry going on at the enzyme active site. And it may help to compare this type of reaction, an enzyme catalyzed reaction, to a reaction that does not take place on an enzyme active site. And here's an example of that that's relevant to what we've been discussing in the course. So this is the reaction of the non-enzymatic glycation of hemoglobin, right? So we have hemoglobin reacts with glucose in the blood to make HbA1c, or glycated hemoglobin, which is shown here. So this reaction, again, there's no enzyme. And if we look at the kinetics of this, so the rate of product formation, the rate of the reaction versus the substrate concentration here, glucose, we see it's a linear rate. It does not saturate, just keeps going up at the same rate, okay, because it's just dependent on the concentrations of glucose in hemoglobin, and there's no enzyme active site. Okay, so no enzymes involved, and you see no saturation kinetics. Now, I want to try an analogy here. I realize enzyme kinetics is not what most people think about. It is not intuitive. The understanding of this mathematics is not intuitive for most people. So I want to try more of a big picture analogy here. And that is of the matchmaker, Yenta. If you remember seeing Fiddler on the Roof, there's a matchmaker. Her name is Yenta. So we're going to call Yenta the enzyme here, okay? And so in our reaction, we're considering, we have the matchmaker, Yenta, with a man and a woman, and she is working together with them in order to catalyze a marriage, right? So we have a product formed here, one marriage, and Yenta now can go on and work with another male and female to catalyze another reaction, okay? So that's our analogy here, okay. Now, if we were going to study how fast Yenta does this, we'd set up the situation, right, to make the math easy, and we'd make sure that Yenta had lots of bachelors. So a number of bachelors would not be rate limiting at all. And we would only alter the number of bachelorettes, right, shown in the pink symbol here. And then we'd measure how fast she can catalyze marriages. All right, by looking at different concentrations, different numbers of bachelorettes to see how does the number of bachelorettes affect how fast this this reaction could occur, the number of marriages, right? And what we would see if she was behaving the way an enzyme behaves is that we'd see saturation kinetics, right? As we increase the number of eligible bachelorettes, the number of marriages per year that Yenta could create would go up as there is more and more bachelorettes until we started saturating her abilities, right? Even if, we could have an infinite number of bachelorettes available, but there's only so much a Yenta can do in a year. Okay, and we could call this the maximal rate at which one Yenta catalyzes marriages. Okay. Now, getting actually measuring like what is the number of bachelorettes that gives you this maximal rate, that's really hard to do because it's slowly approaching that maximal rate. But what we can do is take half that maximal rate, okay, and determine the number of bachelorettes that allows Yenta to work at half her maximal rate. And this is what we often do in analyzing curves like this, is look at the half maximal, okay? So we're gonna see that used a lot here, is that what is the number or concentration of bachelorettes that allows her to work at half the maximal rate? Okay. So how would the situation change if the village recruited a second Yenta, right? A second matchmaker? What would we expect? Well, you'd expect that the maximal rate would double, right? If the second Yenta worked just as quickly as the first Yenta, if they were identical, molecularly identical, then you'd expect a doubling. And that's exactly what we see for enzymes. And the reason why I'm explaining this to you, why it's important, is you're also studying gene regulation right now. And a lot of those genes being regulated are genes that encode enzymes. And the reason that's important is that the level of enzymes matters in terms of the reaction rates that go on in a cell. If you double the level or the concentration of an enzyme in a cell, you double the reaction rate that can occur. Okay, so now let's look at enzymes that work the exact same way, all right? Here is some data from a real enzyme. This happens to be an enzyme called DNA topoisomerase 2 that unknots DNA and unlinks it, and it uses ATP as one of its substrates. So here we're looking at the rate of the reaction compared to how much ATP is provided, okay? And we see it. Again, it saturates, right? And this type of curve that the most enzymes follow was analyzed back over 100 years ago by a team, Michaelis and Menton. It's sort of interesting that Michaelis is a young woman working with a German biochemist. She was a young budding biochemist at the time, working with a German biochemist, working together. And they realized that this form of a curve fits an equation such as this. And now we call this the Michaelis-Menten equation, where the rate of the reaction equals the maximal velocity times the concentration of substrate divided by a constant that we now call the Michaelis constant plus the substrate concentration. Okay, you do not need to memorize this equation. This is the equation that fits these data. And actually, as I was trying to determine Vmax and Km, these were my data. And I used a computer to fit these data to this equation. The line you see here, the curve, is that fit. And I found out the accurate Vmax, the maximal rate, and this Michaelis constant for this enzyme. Okay, and so we're not going to go into those details, but we're going to sort of think about it a little more conceptually and just look at the data, okay, and estimate. So we can estimate, we see saturation occurring here. The rate is not getting much faster with more ATP, okay? And we can see that it looks like it's plateauing somewhere around here where I've drawn the purple dotted line. We'll call that the Vmax, right? And we can get the half of that. So this Vmax 0.48 will go to half of that, 0.24, half the Vmax, right? And if we draw down half Vmax and then draw a line down, what is the ATP concentration at half Vmax? That equals what we call the Km, or the Michaelis constant. Okay, so that's the Km is the concentration of substrate. Here ATP, it's about 5 micromolar ATP. That's the ATP concentration that gives half the maximal velocity. Okay, so again, the Vmax here is about 0.48 micromolar per second. It's a rate. And the Km is 5 micromolar. It's a concentration of the substrate, which here is ATP. Okay, so let's summarize what these terms mean. Vmax is the rate at which a particular enzyme at a particular concentration catalyzes a reaction when it is saturated with substrate. It's the maximal rate for the reaction. Okay. Km is the concentration of substrate required for a particular enzyme to catalyze a reaction at half maximal rate. And it is a constant that does not vary with the enzyme concentration. Whereas Vmax, if we remember back to Yenta, when we doubled the number of Yentas, the maximal rate doubled. Well, Vmax, it changes with enzyme concentration. Okay, so another important thing about Km is that the rate of the reaction is most sensitive to changes in substrate concentration below the Km. So let's explore that for a moment. Okay, so here we're looking at the same data. Now, if we look at substrate concentrations, ATP concentrations that are lower than the Km here, right? So the Km is 5 micromolar. If we look at, say, 1 and 3 micromolar, the difference in rate between 1 and 3 micromolar, the rate almost doubles when we do that small change in ATP concentration. But if we look out here at 30 and 50 micromolar, right? If we increase from 30 to 50 micromolar, we see only a pretty small change in the rate of the reaction. So in order for there to be a big change in the rate of the reaction, the substrates should be below the Km, the concentration of substrates. And therefore, for many regulatory enzymes, particularly those that are sensing the substrate concentration in the cell, they have evolved to have Km values that are higher than the physiologic substrate concentration. In other words, if this is what the curve looked like, the physiologic substrate concentrations would be down here, so that a small change in substrate concentration would cause a big change in the rate of the reaction. Okay, so we'll analyze some examples of those in class. Okay, so the last of the third these kinetic parameters we're talking about is called the kcat, or the turnover number for an enzyme. Right. So in the last video, we talked about the enzyme OMP decarboxylase that's involved in pyrimidine biosynthesis. And we talked about how the kcat, or the turnover number, is 39 per second. Right. And the way I like to think about this is when the enzyme is saturated with substrate, so there's lots of substrate around, each molecule of enzyme can produce about 39 UMPs in every second. Okay, so I think about it on a per molecule basis. I think that's the easiest way to think about it. And therefore, since we're thinking about it on a per molecule basis, the kcat is independent of the enzyme concentration present in a reaction or in a cell. So it's a real constant, independent of concentration of the enzyme. And for example, if we go back to our analogy of Yenta, it's the number of marriages that each Yenta can arrange per year, as long as the number of bachelors and bachelorettes aren't limiting. Okay. So how do we determine the kcat? Well, you can get that from the maximal rate and divide it by the amount of enzyme that was used, the total amount of enzyme used in the reaction. Okay, so whereas the Vmax varies with the concentration of enzyme present, the kcat is independent of the enzyme concentration. So let's practice these. Let's look at this. Okay, so here was our topoisomerase reaction, right? And our Vmax here was 0.48 micromolar per second. We saw that here, 0.48, right? And the enzyme concentration used in the study was 0.1 micromolar. Therefore, this enzyme has a kcat of 0.48 divided by 0.1, which is 4.8 per second. Okay, so now there's a chance for you to practice. Let's look at the enzyme glucose 6-phosphate dehydrogenase. Remember, this is the catalyzes the first step in the pentose phosphate pathway. And if you have a mutation in it, it can cause hemolytic anemia that's induced by reactive oxygen species. Okay, so let's say we have a reaction in which we're doing this in a test tube, right, to analyze how this enzyme works. So let's say we have 0.01 micromolar glucose 6-phosphate dehydrogenase enzyme. There's lots of NADP+, which is one of the substrates, and the following amounts of glucose 6-phosphate. Okay, concentrations, right? So here's our glucose 6-phosphate concentrations, and we measure how fast the reaction is, right? How much product is formed in every second, and these are the values that we see. So just looking at these numbers, can you approximate what the Vmax, the kcat, and the Km and the kcat are for this reaction? Right? You may want to pause the video and see if you can do this on your own. Okay, so the Vmax looks like it's going to be about 25 micromolar per second, right? We see between 200 and 500 micromolar substrate. We don't have a very big increase. It looks like it's plateauing somewhere above about 23. So we'll estimate it's going to be around 25 micromolar per second is the Vmax. So then how do we determine the Km? Well, the next step is to look for the half Vmax, right? So if it's about 25 micromolar per second, 1/2 Vmax is going to be about 12.5 micromolar per second. And Km is a concentration of substrate. What's the concentration of substrate here? Well, it was 50 micromolar. So our Km is about 50 micromolar glucose 6-phosphate. Okay, and what's the kcat? Right? Well, kcat is the Vmax divided by the concentration of enzyme used. And we saw we used 0.01 micromolar enzyme. So the kcat here, we divide this out, is 250 per second. Okay, so now let's practice what changes when we have a change in enzyme concentration and what does not. So our first, let's if you remember correctly, the heme is an important inhibitor of delta ALA synthase, both the transcription and translation of this enzyme, right? So if we have a transient elevation of heme levels in a erythroblast, that's going to result in a decrease in the transcription and translation of delta ALA synthase. Question is, what effect is this going to have on the Km of this enzyme for its substrate succinyl-CoA? It's going to increase the Km, decrease it, or have no effect. Okay, you may want to pause the video here if you want to think about it. Okay, so it's not going to have an effect. So changing the concentration of an enzyme does not affect the Km, right? So remember, Km is an approximate measure of the affinity of an enzyme for its substrate. So, and with lower concentrations, meaning tighter binding, right? I don't think I've mentioned this before. That's another way to think about Km. It's an approximate measure, the affinity of an enzyme for its substrate, and the Km is independent of the concentration of enzyme used, or the concentration of enzyme in the erythroblasts here. Okay, let's try this. Same scenario. What effect is this decrease in transcription and translation of delta ALA synthase likely to have on the Vmax of this reaction? Increase, decrease, or no effect? Okay, you decrease the amount of enzyme, the maximal rate of the reaction will decrease. So Vmax will go down when there's less enzyme present. And lastly, what effect is this going to have on the kcat of the reaction? Increase, decrease, or no effect? This one is no effect. The kcat is independent of the concentration of enzyme. I like to think of it in terms of kcat as the amount of product one molecule of the enzyme can produce per second. Okay, it doesn't matter how many molecules there are of enzyme if you're thinking about how much can one molecule produce per second. Okay, so again, let's put all this together, summarize. Vmax is the rate at which a particular enzyme at a particular concentration catalyzes a reaction when it is saturated with substrate. Km is the concentration of substrate required for a particular enzyme to catalyze the reaction at half maximal rate. This is a constant that does not vary with enzyme concentration. And kcat is the turnover number for an enzyme, right? It's essentially how fast each molecule of enzyme can convert a substrate into a product when the substrate concentration is very high and therefore not rate limiting. And therefore kcat does not vary with the enzyme concentration. Okay, so let's finish off this discussion by considering some common mechanisms for regulating enzymes other than by gene expression. And two of them that you should be thinking about are post-translational modification. Any type of post-translational modification can change the activity of an enzyme in a variety of ways, increase it or decrease it. And then there's reversible binding of activators or inhibitors. And these can be small molecules or proteins, but they can change the kcat, Km, Vmax of an enzyme. So let's look at an example. So muscle glycogen phosphorylase, right? So this is the enzyme we have in our muscles that releases glucose from glycogen, right? It gets activated when our muscles are active and need to use more glucose, right? So the glucose gets released from the glycogen stores. Okay, so here glycogen phosphorylase is a homodimer, two identical subunits. Here's the form in which it is inactive. It's just sitting there in our resting muscles, not doing anything. And it can be activated in two different ways. One is if there's increased levels of AMP in the muscle cells, right? So ATP starts being used by the muscles and ends up creating more AMP, right? And so there is a allosteric binding site for AMP separate from the active site of the enzyme that changes the conformation of the enzyme and puts it into an active conformation. So AMP is an allosteric activator, and this form of the enzyme is active. There's a totally different way of also activating the enzyme, and that's by a kinase. It's called phosphorylase kinase. It's the kinase that regulates glycogen phosphorylase, hence the bizarre name, right? And it's a kinase, so it uses ATP to transfer a phosphate onto a particular amino acid in the phosphorylase of the phosphorylase. So now we have phosphorylated glycogen phosphorylase, which is also active, right? So it's two different ways of activating. And actually, to get the most active form is when there is both activation of phosphorylase kinase and lots of AMP in the muscle cell. This form is the most active form and very rapidly releases glucose, phosphorylated glucose from glycogen, right? So how does phosphorylase kinase get activated? Well, one way is by increased calcium inside the myocyte cytosol, right? That gets released from the sarcoplasmic reticulum, and the calcium calmodulin ends up activating phosphorylase kinase. Another way is if you have increased epinephrine. and which activates a G protein coupled receptor, increasing adenylate cyclase activity, producing more cyclic AMP, which binds to protein kinase A, the regulatory subunits, causing those to fall off. And we have activated protein kinase A, which phosphorylates kinase, right? So a long story putting some physiology together with this kinetics. That here's an example where we both have an allosteric activator, AMP, as well as a post-translational modification, phosphorylation, affecting the activity of this enzyme. Okay, and so let's finish off looking at some enzyme inhibitors. And these can be both naturally occurring and pharmacologic. There's lots of small molecules, feedback inhibitors of a lot of metabolic pathways can inhibit enzyme activities, as well as there are pharmacologic, a lot of drugs are enzyme inhibitors. And one type of enzyme inhibitor that you should recognize is a competitive inhibitor. And what that means simply is that it's usually that the inhibitor binds at the enzyme active site and it competes with the substrate or substrates for binding, right? And in thinking about this competition, should think about the fact that, if there was high enough substrate provided, the substrate could compete away any inhibitor, and the enzyme could still achieve the same maximal rate of reaction. Right? So the Vmax doesn't change. There just has to be a lot more substrate present to get to that Vmax. And that's part of the definition of a competitive inhibitor. That's how we analyze inhibitors to determine if they're competitive inhibitors. So let's look at an example of that, right? So, right, if this is our substrate versus rate curve of an enzyme with no inhibitor present, how would this curve change if we added a competitive inhibitor? Think about that, maybe draw it out what you would expect to see. So it should look something like this, right? It doesn't change the shape of the curve. It just means that there has to be a lot more substrate present to compete with the inhibitor to reach essentially the same Vmax. Right, so the Vmax doesn't change, just need a lot more substrate present to compete. And if we're going to look at the half Vmax, we would see that the Km with the inhibitor is going to be at a higher substrate concentration. It's going to take more substrate to get to that half maximal velocity when the inhibitor is present. Now, back before everybody had computers to fit their data, these types of data were often, the equation for the Michaelis-Menten equation was often converted to a linear form. And one of the most common ones was a double reciprocal plot format, right? So we could draw it as a line and use a ruler and graph paper. So what does that look like? So here's our Michaelis-Menten equation. Now, double reciprocal means we're just taking the reciprocal of both sides of this equation. So we have 1 over the velocity or the rate of the reaction equals Km over Vmax times S plus 1 over Vmax, right? And the nice thing about this is it's in the format of Y equals mx plus B, which is the equation for a line. Okay, so this is what that looks like if we plot it out. So we have 1 over the velocity versus 1 over the substrate on the x-axis here, right? The intercept here, B, is 1 over Vmax, and the slope of the reaction is Km divided by Vmax. All right, so what would it look like if we added a competitive inhibitor? Well, key thing is Vmax doesn't change. So 1 over Vmax also does not change. And this is what it ends up looking like with a competitive inhibitor. So in some board prep books, study materials, maybe as an undergrad, you learned this form. Again, the reason people really did this is because they didn't have computers in the past, and you could draw lines and use rulers, and you'd have data without the inhibitor, and then there'd be data points here with the inhibitor, and you would see digital lines cross. And if they cross right or where the lines cross, and when they cross right on the y-axis, then you could be confident that it was a competitive inhibitor. Okay. So how about for the other types of basic inhibitors, non-competitive and uncompetitive inhibitors? And I'll just say that the distinction between these two types of inhibition is rarely important, and I would say generally not worth worrying about. Okay. In both cases, the inhibitor binds to the enzyme somewhere other than the active site, and therefore does not compete with the substrate for binding, right? And often these are allosteric inhibitors, right? So binding elsewhere, inhibiting the structure, the rate of the enzyme through stabilizing a different conformation of the enzyme most commonly. And the result is always that the Vmax is decreased by either of these types of inhibitors. And part of the distinction is whether the Km is affected, and they may or may not be. And I don't think that's so important for the mechanism. Okay. So I just want to, as we finish, touch back on one thing we talked about in the prior video, and that is this transition state theory of enzyme catalysis, right? Which was that the enzyme active sites evolved to be most complementary to and have the greatest number of binding interactions with the transition state of the reaction. So if you think about it, therefore, what effect should a chemically stable analog of a transition state have on enzyme activity? When we say a chemically stable analog, this is a molecule that it's similar in structure to the transition state, but it's inert. It does not fall apart. The enzyme doesn't react with it. It just has all the binding interactions that are like the transition state, right? Well, these are extremely tight binding competitive inhibitors. They bind at the enzyme active site, so they're competitive inhibitors. And they are so perfectly aligned to the structure of that enzyme active site that they bind incredibly tightly. And one reason people care about this is one way to think about how you might design a drug to inhibit a specific enzyme, is to think about what is this transition state of an enzyme catalyzed reaction. Okay, so let's summarize. The rate of reaction is most sensitive to the substrate concentration below the Km. Okay, we talked about that in terms of Km. Rate of reaction is most sensitive to the substrate concentration below the Km. Competitive inhibitors increase the apparent Km for the substrate, but have no effect on Vmax. Okay, so the competitive inhibitors. A change in enzyme concentration affects its maximal rate Vmax, but not its Km. Okay, the substrate concentration at half maximal rate, or the Kcat, the turnover number. And many enzymes are regulated by allosteric effectors, by mechanisms quite similar to the regulation of oxygen affinity for hemoglobin. Okay, thank you for watching.